We'd like to welcome our presenter, Dr. Dritan Agalliu. He is an associate professor of pathology and cell biology and neurology at Columbia University's Irving Medical Center. He is a neuroscientist with 20 years of research experience in developmental, cellular, and molecular neuroscience, and has specialized in the interaction of brain and blood vessels in healthy and as well as diseased central nervous systems.
The questions that we are discussing today were asked during our live webinar from earlier this year. The role of adaptive immunity in vascular and neuronal dysfunction in post-infectious autoimmune encephalitis. A recording of that webinar can be found on the PANDAS Physicians Network website. Thank you for taking the time to meet with us and answer these questions.
Thank you, Susan, for inviting me back. I do apologize that I was never able to write an answer to the questions from the audience, but I thought this was a better way to basically remind them about our findings and then answer live questions that people had on the webinar. Thank you.
Terrific. Well, we're glad you're here today. I'll start by reading off a question and then we'll let you just take it from there, okay?
Absolutely.
Okay, so you described in your talk the identification of cytokine and chemokines from the sera of PANDAS and PANS patients that were low or absent in controls. We had a number of questions about the controls. The first was, were the healthy controls ever infected with group A strep? And the second was the anti-group A strep antibody reactivity similar between the groups?
Excellent question. In order to answer this, I'm going to share with you one of the slides from the presentation, just to remind the audience about our findings and then answer the question. Basically the question relates a little bit to the findings that is presented here, where we found that indeed the sera from children who suffer from PANDAS and PANS, whether they were girls or boys, significantly upregulated the transport of protein like serum albumin across an in vitro model of the human blood-brain barrier in culture, but the controls didn't. And this was even higher compared to the exposure of the cells to some of the inflammatory cytokines, IL-1 beta and TNF-alpha. And of course, given that group A strep infections are common in the childhood, I think the question is really highly relevant, whether indeed these children had group A strep infections. We don't know the answer to that question. I think we assume that the children who were healthy had some group A strep infections. However, in the interviews of these children, they didn't remember to have any severe episodic of like strep throat. However, it's impossible to exclude the possibility that these children could have had milder forms of strep throat or group A strep infections, since they happen very often in children. To answer question number 2, that whether indeed the antibodies against group A strep were present in the control sera or not, we did not test for the presence of group A strep antibodies in control sera. But however, to answer the question, is a one experiment that we did to prove that the effect that we saw on the sera was mostly due to cytokines and chemokines and not the presence of antibodies, was that we depleted the antibodies present in the sera from either control or PANDAS children using biochemical approaches. And then we exposed the sera that were depleted from antibodies to the culture of endothelial cells, and we found similar results. And so this suggests to us that, in fact, the effects that we saw on the blood-brain barrier were predominantly due to the presence of the cytokines and chemokines, and are highly unlikely due to the presence of antibodies. In fact, in this cohort of patients, when we measured the binding to the dopamine receptors, D1 and D2, we didn't see a huge difference between the control and PANDAS and PANS patients, in particular this cohort, suggesting that indeed the effects that we saw were most likely associated with this abnormal profile of cytokines and chemokines.
Great, thank you. The next question is around ocular issues, about why do you think PANDAS and PANS children experience ocular issues and whether or not that might be related to Th17 cells?
So the retina is part of the central nervous system. And, you know, in other autoimmune diseases of the brain, such as multiple sclerosis, there are, in fact, ocular issues. In fact, there is a form of MS called neuromyelitis optica, where, you know, the body generates antibodies against a protein called aquaporin 4 that is expressed in the glial cells that surround the blood vessels and are important for forming the blood-brain barrier. And so these antibodies that are generated disrupt the blood-brain barrier and cause predominantly, you know, problems with vision. It is possible that, so we anticipate that some of these cytokines that we identified in the sera from PANDAS or PANS patients have the ability to break down the blood-retina barrier, which is very similar to the blood-brain barrier. So it's possible that some of the ocular issues are related to a similar damage that could happen in the brain. Obviously, you know, another explanation could be that in these children who present with ocular issues, they may have antibodies that target the eye rather than the basal ganglia. And, you know, it would be very interesting, of course, to explore this further in the future.
Okay. The next question is, what is the relationship between Th17 cells, the antibodies getting into the brain, and the activation of the microglia?
That's an excellent question. So I'm going to share that by basically discussing one more time the slides from the presentation, because I think that makes it makes my life a lot easier. So as I said, one of the things that we found from our animal models for basal ganglia encephalitis is that after multiple infections with group A strep, we generate population of T cells called Th17 lymphocytes in a lymphoid tissue found in the nose called the nose-associated lymphoid tissue that is equivalent to the tonsils in the humans. And what we demonstrated in this study is that we think that likely the Th17 lymphocytes travel from the nose into the brain along the olfactory route, so following the route of the olfactory sensory neurons that are important for the sense of smell. And then once in the brain, we found that indeed Th17 lymphocytes secrete specific cytokines and chemokines that can damage the blood vessels in the brain to allow then antibodies to enter the brain. In addition, in this model, we also found that the immune resident cells of the brain called microglia become hyperactivated and inflamed, causing a sustained inflammation. And so we do think that, and this is also characterized with loss of neuronal activity. Moreover, we do know that indeed, if you remove Th17 cells in the mouse, then you get basically protection of the blood-brain barrier, and then you don't get activation of microglia. So at least in our animal studies, we do know that the presence of the Th17 lymphocytes is essential for the rest of the pathology.
I remember when some of this research came out, I thought it was incredibly fascinating. Thank you for sharing that with us again today.
Absolutely. Thank you.
Okay, so the next question is, can you say anything about how the B cells might be interacting to influence the blood-brain barrier in your mouse model? What are the B cells doing in the mouse model? It looks like the healthy individual serum also triggered some response in your cultured cells.
Right. So basically, we haven't really, in our mouse model, this is a very good question, because in the context of autoimmune encephalitis, B cells do play an important role because they are the cells that generate the antibodies. And also in children with PANDAS and PANS, B cells play an important role. B cells play an important role also in diseases such as multiple sclerosis because they are critical for generating antibodies that target the central nervous system. So in our animal model, we have looked carefully and we do find B cells present in the surface of the brain in a structure called the meninges. So if I share that last slide, I can point out where we find them. So we do find the B cells here at the surface of the brain, but we do not find them being present inside the brain. So therefore, we do know that B cells per se are not generating antibodies inside the brain. And these antibodies have to somehow enter the brain from the blood vessels because the B cells are not there. When we published this study in 2020, we did perform one experiment where we depleted the mice from B cells. However, under those conditions, mice were not able to survive well the infections. And so it was really difficult to assess, what was the contribution of B cells for the disease, because obviously when you don't have the antibodies, you can't generate a proper immune response to fight the infection. So therefore, I think the role of B cells in our animal model is still a bit of an open question. And I think some of the studies that we're doing right now with where we're isolating all the cells of the brain from the mouse and look at their changes in the gene identity and expression are going to be important to see whether indeed B cells are there, and if they're there, what are they producing that could be relevant.
We'll look forward to that research.
Yes.
We'll have to do another call when you have that ready.
Absolutely.
All right. The next question is, can the olfactory route of coronavirus be a relevant cause of causing central nervous system inflammation, autoimmune encephalitis? I think generally asking about how COVID-19 might be impacting this patient group.
Absolutely, and that's a great question, because has been a question for us in our minds for actually a very long time. I mean, I think since COVID started in early 2020, we were really intrigued, because obviously the neurological sequelae of COVID have been seen actually in increasingly in the adult population, but also right now some of them are being seen in the pediatric population. And obviously with the presence of the neurological sequelae, I think one of the questions that arises is, how are these caused? Are these caused by the virus entering the brain, or are these caused by the secondary effects? As you probably know, and I think everyone probably knows, is that indeed the virus causes the what we call a cytokine storm, so the production of a large number of cytokines and chemokines that are also, they cause very severe illness and hospitalization. And so to address this question, I think in the early 2020, I was part of an investigation that of a study that was conducted here at Columbia, where we analyzed the brain pathology of 41 patients who unfortunately died from COVID from the period of April 2020 until June 2020. This was the time when the COVID pandemic was extremely severe here in New York City, and we had a large number of cases and also deaths here at Columbia. And the surprising finding from this was that indeed when we examined 20 different regions of the brain to look for the presence of the virus, and also to look for the presence of immune cells in these regions. And the surprising bit was that when we looked on the olfactory bulb, which is the area of the brain that is very close to the nose, we didn't see the presence of the virus. In fact, throughout the brain, we saw very little virus present in the brains of these patients, compared to like the higher titer that you see in the nose, for example, and obviously in the lungs. And I think the data to us clearly indicated that even though it is a possibility, it's very unlikely that the virus enters the brain through the olfactory route. Now, there might be cases where it may happen, but it's not a common way by which this virus can cause brain disease and neurological sequelae. The most likely cause is that indeed the cytokine storm is the predominant mechanism by which it causes vascular damage and also brain pathology.
Well, I appreciate you're doing that work, and also just want to express my appreciation to those patients and their families that were so generous to help us with that understanding. So
yeah, and if in case the audience is interested, that paper is already published in Brain early in 2021.
Okay, terrific. I'll make sure we have a link to that on the PANDAS Physician Network site as well, so that people can find that. Thank you. Okay, the next question is back to group A strep. Your mouse model looks at the implications of group A strep. What other infections and immune stimulating situations or events are known to disrupt the blood-brain barrier and impact the central nervous system? And are you looking into something similar in terms of impact with other bacteria or viruses?
That's a very good question. Yeah, other infections obviously are known to disrupt the blood-brain barrier and impact the CNS. And I think the most known cause that also has been associated with PANDAS and are infections with Borrelia burgdorferi and other Lyme's that, and other bacterial agents known to cause Lyme. Obviously, the mechanisms are slightly different because unlike group A strep, which doesn't infect the brain directly, but causes an immune response that targets the brain, I think infections with Borrelia burgdorferi and some other Borrelia species that cause Lyme disease have been found to cross the blood-brain barrier and enter the brain. And obviously, this is something that we're interested in terms of trying to identify whether indeed there are similarities in terms of the cytokines and chemokines that are present in children who suffer from PANDAS and PANS that were infected with either group A strep or with Lyme. Because, potentially if we consider developing therapeutics that target this cytokine storm, it would be beneficial to know whether indeed this could be specific for a subgroup or could be broad or for all children who suffer from PANDAS and PANS. But obviously, there are a lot of other agents that can disrupt the brain barrier. I just brought out one example here.
But quite a few out there that we've already have research on that we can talk about another time.
Absolutely.
All right, so the next question is, what about children, adolescents, and young adults who experience symptoms from multiple pathogens? Is there a cumulative effect, or is it really the same impact that they have on those children and adolescents?
I mean, certainly the effects could be cumulative. For example, in the study in 2016, when we analyzed for example, the infections with other pathogens, we did see that, I mean, even though group A strep was the most robust in inducing the generation of the Th17 lymphocytes, staph also can generate Th17 lymphocytes. And so even it's unclear whether these T cells will go in the brain. So in other words, if you if a child has had multiple infections with group A strep and has already generated a robust immune response, including T cells, and then gets infections with another pathogens like staph that can induce also a robust immune response and generation of T cells that can have a exacerbation effect. Obviously, some of the viruses also are known to trigger a T cell mediated immune response. And so, in some cases, they could have an, compound effect. But I think it's difficult right now to know, obviously. But the fact that, there are a lot of, relapses in children who suffer from PANDAS and PANS suggests that, that children and young adults are exposed to multiple infectious triggering agents, and some of them do the same thing. They activate the immune system. And so in a situation where your immune system is, there is an imbalance in the immune response that can be exacerbated even further.
I think that really leads us into the next question, which is, you've got mouse data and human genetic data, and both indicate that PANDAS and PANS is a neuroinflammatory disease. What else? There's obviously a lot of research that's still needed in many different areas, but if you were to think through what else is needed for these disorders to be universally recognized and for these patients to be treated, what would you suggest is really needed as next steps?
So obviously our findings in the mouse study and in the with a small cohort of patients do indicate clearly that this is actually a disease that is neuroinflammatory in nature, because obviously a lot of the genetic risk factors we identified from whole exome sequencing in patients were in genes that regulate the immune response, the adaptive immune response. However, our data were collected from a very small number of patients. And so I think in order for the scientific and medical community to really embrace a immune mechanism driving disease in PANDAS and PANS, you need a larger number of studies. Obviously, for diseases like schizophrenia, for example, bipolar disorders, there have been larger consortium. Autism, there have been a lot of large consortia genetic studies that in order to identify genetic risk factors. And I feel that that is one way to go forward, to basically have a larger genetic study to with a big established cohort from multiple well-established centers to address this. I think this is also the second one is really kind of trying to develop approaches to build a large repository for the patient, a database that is connected with blood and sera, as well as the genetic, because I think this is critical in order to conduct studies. What are the biomarkers for disease? And I feel right now everybody is operating with a small pool of patients that have identified locally, but there is really a big missing piece, which is building a large repository. And I think that this is really critical because if you want to think about moving the research forward, it's really difficult to do that if you are trying to do active recruiting all the time, because you can recruit only a very small number of patients at any given time. And right now, for example, when we're still under the COVID-19 pandemic, the recruiting is even harder because, the hospital center have switched to telehealth. And so bringing patients in the clinic, it's a little bit trickier. But I think if we want to really try to understand the link between the disease, genetic risk factors, and then to identify potentially biomarkers, we need to build a large repository. But I still think, even though in our study we identified a few cytokines, we don't know if these are true biomarkers of the active disease. Because I think, future studies would be needed to be done where we do a larger, the cytokine profiling in a large number of patients at various stages of disease, in acute and then in the remission phase, to really determine if the profile of this abnormal cytokine profile that I discussed is actually changing. In other diseases, including multiple sclerosis, there's a lot of evidence that in the aberrant composition of the gut microbiome influences the disease significantly. And this is a big area that is still unknown in the PANDAS field. There is a small study done in Italy suggesting that children who suffer from the PANDAS and PANS and are known to have a lot of gut abnormalities have an abnormal microbiome, but that study is, done in a small cohort, and now we have the possibility to do, we do have the tools to do a much larger study, and I think this study would be beneficial for the community as well, and potentially for building future therapeutics.
Okay, great.
There are a lot of ideas.
Yeah, it's funny, the next question that was asked was just simply, do you feel there's opportunity for collaboration between researchers? And I think you've just answered that question, that there's a lot of opportunity, right? Yeah, the seed research is there. It's now time to increase our sample size and to work across sites so that we can see patients at different times.
I think that is important. And I do think that there is actually a role, not just for the scientists and clinicians to collaborate, but also nonprofit organizations and advocacy groups to really help with building robust repositories that potentially could be useful, not just to have a database of patients, but potentially to build repositories that you could also create IRBs that you can collect samples that are stored so that investigators can use these samples for their studies, because the study can be done much faster if these samples are already collected rather than, waiting to be collected. I mean, there are some studies that can, but others can't.
So how would people that are listening to this help make that repository or your research move forward?
Well, it's I think a big effort is donations, if possible. I do think that there are also other ways. Also advocacy. I think it's also another important thing. And we do also because I think it's important for us to have the PANDAS and PANS in the map, right? And that people, even though there is a lot of controversy, it's important to discuss the controversy. It's important to put the disease in the map. It's important to discuss the disease in the forums of rare diseases as well, because there are opportunities for rare diseases to get some funding to build repositories. In fact, today, the CZI initiative, there was a CZI initiative to build a repository for rare diseases, even though practically a lot of the parents think that PANDAS is more prevalent. I mean, I think when you think of larger pediatric diseases, these are relatively rare diseases. And I think we could benefit all from really, putting a lot of effort out there to put the disease in the map. And then from there, we could build ways to request funding for rare diseases, but as long as the disease is recognized.
Okay, terrific. Going back to the question about collaboration, is there anyone you hope to work with in the future?
Yeah, so one investigator that I've been really excited to collaborate, I think obviously, I've been trying to do a lot of setup collaboration with Jennifer Frankovich and Kyle Williams, particularly for our genetic studies, because obviously, it would be important to create a large consortium of patients that we can address, what are the genetic risk factors for the disease. Right now, we have a very small population, and I know that Jenny Frankovich is doing a study at Stanford already, but I do think this is where the importance of the consortia comes into play. And also in terms of the mechanisms of disease, I'm very excited to potentially collaborate with Gary Westbrook at OHSU, who has done a fantastic mouse model for autoimmune encephalitis, because there are a lot of similarities in the mechanisms of disease, and exploring those, and in particular, the relationship with blood-brain barrier images is really critical. I think there are also other investigators, but one of them that I'm also excited is an Italian investigator who is right now in England, and his name is Fulvio D'Acquisto. And I think he's on this really highly relevant protein that is secreted by T cells, which causes obsessive compulsive disorders and mood disorders. Whether that is relevant for our animal model would be really, the protein is called imudin, and I think I'm very excited to see if that would be also relevant in our animal studies if we are to perform our infectious model in mice lacking this protein. I can go on, but I think there are a lot of people that I'm pretty excited to collaborate. And obviously, I think the repository would be something that would be really helpful for all of us to collaborate for the benefit of the community.
I think everyone would be very interested in seeing that. I appreciate your time today. I know we all do. You bring such a passion for your research, and we know you're busy doing that work, but you're also so generous in your time and translating that to the rest of the community, whether they're researchers, physicians, or parents of patients who are suffering. We hope you've gained something from this information and just deeply value your time.
Thank you so much, Susan, for inviting me. It's always a pleasure.
Have a wonderful night.
You too.